Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 4 principles of inheritance and variation. In that we are going to see inheritance of two genes. So before going into the topic, watch the full video children. Don't skip the video. It is for your good only. Yes. If you have any doubt you can ask me in the comment box also. I'll reply to you immediately. Yes, let's start. So Mendel, he studied the inheritance of one character at a time, which was known as monohybrid cross. Then he became curious to know whether two different characters are inherited together or independently. So in order to answer this question, he performed a dihybrid cross. So we are going to see dihybrid cross and linkage in this session. Yes, let's start with the topic dihybrid cross. So what are the parents used here? So Mendel, he selected two true breeding pea plants. True breeding pea plants. So parent one round and yellow seeds. Parent one was round and yellow seeds. I'll show you round and yellow seeds. Yeah, can you able to see here? Round and yellow seeds. And then the genotype. What is the genotype? You can able to see capital RR and capital YY. And then parent 2 wrinkled green seeds. And the genotype, what is the genotype? Small rr and small yy. Yes. So since these plants were pure breeding, they produced only one type of gamete. What is the one type of gamete produced here? So this is the one type of gamete. Yes. So after fertilization, what happened? So all F1 plants have round yellow seeds. So this is the F1 generation. So why all F1 plants were round and yellow? See, it is because of the round. Here, the round is there, no? R is dominant over the wrinkled. And the yellow here, this Y, is dominant over this green. That's why. So exactly the same dominance was observed earlier in our monohybrid crosses also. Then what he did the selfing. He did selfing of this one. Then he observed that seed color segregated in 3 yellow is to 1 green, and also the seed shape segregated in 3 round is to 1 wrinkled. Yes. So what is the ratio over here? It was in the ratio of 9 is to 3 is to 3 is to 1. Can you able to see all these things? Yes, all these are, yes, yeah, this color, all these 9. Now, what is the, all they are, round is dominant over here. Yeah, round is dominant, and also yellow. They are round yellow. Yes, and this one, 2, 3. How they are? They are wrinkled. They are wrinkled and yellow. They are wrinkled and yellow. And these things, this pink one, 2, 3. How they are? They are round and green. Yes. And this only one. This one is the wrinkled green. Yes. So, what you have to remember is, say parents. What are the parents? So these are the parents. Parent number 1 and parent number 2, and F1 generation all were round yellow, and each individual trait still it follows the 3:1 ratio. Let's move on to the next one. So here we are going to see law of independent assortment. So what Mendel observed in his F2 generation, see when Mendel studied both the characters together, he obtained 4 different phenotypes. He obtained 4 different phenotypes. What are the 4 different phenotypes? One was like a round yellow, and another was round green, wrinkle yellow, and wrinkle green. So these appeared in the famous ratio of 9 is to 3 is to 3 is to 1. 9 is to 3 is to 3:1. So each character here independently it follows, like a round is to wrinkled is 3:1. Yellow is to green is 3:1. So when you multiply both the things, what happened? When you multiplied these, you got 9:3:3:1 ratio. So what Mendel concluded? Mendel concluded that when two pairs of traits are combined in a hybrid, yeah, or we can say when two pairs of contrasting traits are present together, the segregation of one pair of allele is independent of the segregation of other pair. Yes, can you understand? So this one he called as the law of independent assortment. And formation of four types of gametes you can see here. What are the four gametes? See the genotype of S. What is the genotype of F1 generation? Genotype of F1 generation was R, capital R, and then small r, and capital Y, and small y, right? So during meiosis, what happened? R it separated from this R separated from this R, and then Y it separated from this small y. So since both segregate independently, four gametes are formed. What is the first gamete? So this is the first gamete, and this is the second one, and the third one, and the fourth one. Yes, children. So each is produced in 25 percentage of frequency, 25 percentage of frequency. So next we are going to see, when this four male gametes, they combine with the four female gametes. When this four male gametes, when it combines with the four female gametes, what will happen? So 4 into 4 in the Punnett square, if you see, means it is a total combination of 16. How many? 4 into 4 is 16 boxes, right? So phenotypic ratio is 9:3:3:1. Yes. So what you should remember for your exam point of view is four gametes are formed. Yes, four gametes you can see, and 16 Punnett square boxes, 16 Punnett square boxes, and also the phenotypic ratio is 9:3:3:1. So next we are going to see the chromosomal theory of inheritance. Although Mendel he published his findings in 1865, scientists ignored them nearly how many years? Nearly for 35 years. And what is the reason behind it? See, there may be a communication gap between the scientists, and also genes could not be physically seen. It was not seen now because the electron microscope was not there during that time, and many biologists believed the inheritance was due to blending. That is a thing. And then Mendel he used mathematics, which was unusual in biology. And then rediscovery, when it happened in 19,000. Rediscovery happened in the year, yes, 19,000. So what happened in 1900? See, three scientists did independently, they discovered the Mendel's work. Who are they? First one is the Hugo de Vries, and then next is Carl Correns, and then Eric von Schermark. Yeah, Eric von Schermark. So what they said, so they discovered, rediscovery was done on his findings. Yes. So then what happened? Discovery of the chromosomes happened. Discovery of the chromosomes. So improved microscopes, it allowed the scientist to observe the chromosomes during cell division. We very well know only during the cell divisions only the chromosomes will be formed. Till then it will be in the form of a chromatin reticulum. Right, children? So next, what scientists they noticed that these chromosomes they occur in pairs. Can you able to see here? Yeah. So these chromosomes they are occurring in the pairs, and then they separate during the meiosis. So it's in the meiosis, see they got separated during the meiosis. They getting separated, and then they enter the different gametes. So here they got separated. End of this meiosis too, they got separated, and they are entering like separated during meiosis, and they enter the different gametes. How exactly like Mendel's factors. Yes. Then Sutton and Boveri. See, Walter Sutton and Theodore Boveri, they compared the chromosome behavior with the Mendel's observation. What they did, they compared the chromosome behavior with Mendel's observation. and they proposed chromosomal theory of inheritance. What they proposed? Chromosomal theory of inheritance. So genes, according to that theory, genes are located on chromosomes. Genes are located on chromosomes, and the chromosomes are responsible for transmitting the hereditary characters. For example, let us take this is our chromosomes, and he said the genes are located on the chromosomes. So this is a gene, yes. So genes are located on chromosomes, and chromosomes are responsible for transmitting heredity characters. You can have a question like, can chromosomes explain why alleles separate during meiosis? Yes, of course, it can explain. And then what is our exam point of view is genes are located on homologous chromosomes. Genes are located on homologous chromosomes. So what happens during meiosis? These homologous chromosomes, they pair and they segregate into different gametes and they carry their genes, that is alleles with them. And exactly as the Mendel's factor, it was explained by Sutton and Boveri. And next we are going to see the chromosome and the genes, the parallel. So whatever we saw in our previous slide, yeah. So let's have one quick look. So comparison. When we talk about these chromosomes, how the chromosomes, they occur in pairs, right? And they separate during meiosis, yes. And only one chromosome enters each gamete. We saw in that diagram part. And then different chromosomes, they the pairs assort independently. What happens? Different chromosome pairs, they assort independently. So genes show exactly the same behavior. Genes also show exactly same behavior. And what he concluded from that? They concluded that since this chromosome behavior it exactly matches the gene behavior, gene must be present on chromosomes. So it became a very strong evidence. See, it supported the chromosomal theory of inheritance. It supported the chromosomal theory of inheritance. And also what you should remember from this, see, behavior of chromosome is equal to behavior of genes. Very important, yeah, is there here. So behavior of chromosomes is equal to behavior of genes. Remember this, don't forget. Let's move on to the next one, Morgan and the fruit fly. So although Sutton and Boveri they proposed the theory, experimental proof was only given by Thomas Hunt Morgan. Yes. So why he used this Drosophila? You would have used some other things also. No. Why he used Drosophila? See the fruit fly. You can able to see the picture of this fruit fly here, children. Yes, the fruit fly. Drosophila melanogaster. It was ideal because it life cycle is only 2 weeks. Yes, life cycle is only about 2 weeks, and also it produces hundreds of offsprings, and it's very easy to culture. And males and females can easily be distinguished. Can you able to see this is the male and this is the female? Yes, and many visible mutations also can be viewed. And Morgan experiment, it firmly established that genes are carried on chromosomes. Where genes are carried on chromosomes. Yes. So you should, for exam tip, you should learn Morgan experiment is equal to the experimental proof of the chromosomal theory. Yes. Let's move on to the next one. So next we are going to see the linkage. uh, some unexpected observation was found by Morgan. See, Morgan expected the F2 ratio to be 9 is to 3 is to 3 is to 1. What he expected? He expected it to be 9 is to 3 is to 3 is to 1. But he observed something different in this. So parental combinations appeared much more frequently than the expected ones. What happened? Parental combinations, yes, appeared much more frequently than expected. So what is the reason for this one? See, the two genes were located on the same chromosome that we know. So they tended to travel together during inheritance. So this phenomenon we called as the linkage. Yes, the chromosome, they tended to travel together during inheritance. So this phenomenon is called as linkage. And what is the linkage? See, linkage is the tendency of genes located on the same chromosome. Tendency of genes located on same chromosome to be inherited together. And what was the result? See, the linked genes, they do not assort independently. That is why the F2 ratio, it deviated from 9 is to 3 is to 3 is to 1. Yes. Yeah, let's see this linkage, that is cross A tight and West cross B loose. Yeah, let's have a quick look. So here, let's see this cross A first. Can you see, children? So here what happened? The parental generation. So this is the parental generation. Let's take the female. So female, it was yellow body and white eyes. Can you able to see? So this is the parental, yellow body and white eyes. And male fly, it was a wild type. You can able to see the genes also. Yes. So here the yellow Y gene and white W gene are located very close. Can you able to see? Yeah, they are located very close together on the same chromosome. So what happened in the F1 generation, that is after crossing, all the female offspring, it became heterozygous. It became heterozygous. And it is not homozygous. Yes, it became heterozygous and show the wild type because the normal alleles, yeah. So these are this normal alleles here, they are no. Here you can able to see this one normal alleles, they are dominant. So the male offspring, we show the recessive phenotype because males have only one X chromosomes, right, children? And then gamete formation. See, here in the gamete formation, see genes are very close together. So crossing over rarely occurred here, right? Genes are, you can able to see how these genes are there very close together, right? So because of this, crossing over rarely occurred. And because of this rare crossing over, 98.7% of gametes are parental types and only 1.3 percentage are recombinant types. Yes, 98.7% of gametes are parental types and only 1.3 percentage are recombinant types. So what this indicated? This indicated very strong or tight linkage. This indicated very strong or tight linkage. And what happened in the F2 generation? See, most offsprings, they resembled the parents. Most offspring, they resembled the parents. Only a very small number showed a new combination. So what it said at last, the parental combination greatly outnumber the recombinant combination. Yes, parental combination, it greatly outnumber recombinant combination. So what we can conclude from this, see the genes are very close together. So recombination is extremely rare. And the tight linkage, this is the tight linkage, right? So the is equal to the low recombination frequency. What is the low recombination frequency? It is 1.3 percentage. Yes, children? Yes. Next, let's see the cross breed. Cross B, it is loose linkage. Yeah, as an example of a loose linkage. Here, the parental generation, let us take this the female fly. It is white eyes, miniature wings, and the male fly is wild type. Yes. So in this cross, so when it is crossed, yeah, when it is crossed, what happened? The white and miniature wing genes are on same chromosome but are farther apart. Can you able to see? Is it like this? Yeah, is it this one like this? No, no. They are very close to each other and they are farther apart. Yeah, far away from each other. And then what happened in the F1 generation? The offspring became heterozygous. The offspring became heterozygous. And what happened? Mostly it showed the wild type. It showed the wild type because dominant allele, they mask the recessive ones. Dominant allele, they mask the recessive ones. And then gamete formation. In the gamete formation, see, these genes are farther apart, right? So crossing over occurred more frequently. It was not like this one now, cross A. So here the crossing over occurred more frequently. Therefore, how the parental ratio was? So 62.8 percentage parental gametes were there, and also how many recombinant gametes were there? 37.2 percentage of recombinant gametes were there. So this indicates the loose linkage. And then what happened in the F2 generation? In the F2 generation, see, many offspring, it showed new combination. You can able to see here many offsprings. Yeah, it showed the new combinations of traits because recombination occurs frequently. So what we can conclude here, see, genes located farther apart on the chromosome, they separate more often during the crossing over. We can say like a loose linkage is like a high recombination frequency, that is 37.2 percentage. Yeah, let's have a quick comparison of this cross A and cross B. They may ask you in examination, compare, yeah, compare the linkage of tight and yes, loose one, right? Loose linkage. So let's talk about the feature also. So gene studied. The first feature is gene studied. So what is the gene studied here in the cross A? It is a yellow and white. And what is studied in cross B? It is white and miniature. And the distance between the gene here is very close. And the distance between the gene here is far apart. And here, what is the type of the linkage? Yes, it is a tight linkage. It is a here it is a loose linkage. And here the parental type is 98.7 percentage, and here it is 62.8 percentage. And here the recombinant type is 1.3 percentage, and here it is 37.2 percentage. Here the crossing over is rare. Here it is frequent. Yes. You may have a question, why does the recombination differ? Yes, why it differs? See, the chance of crossing over, it depends on the distance between the gene. The chance of crossing over, it depends upon the distance. You can see the distance over here, distance between the gene. If the genes are closer, if the genes are close together, crossing over rarely occurs. So low recombination, that is why low recombination. If the genes are far apart, so crossing over occurs more often. Yeah. So high recombination, high recombination. You can see the percentage also. And what is the importance of this experiment? See, Morgan experiments proved that genes are physically located on the chromosomes. That we know. Like genes are physically located on chromosomes, and genes are on the same chromosomes, they show linkage. Yeah, you can, this is the same chromosome only, no? So here one gene is there, here one gene is there. So genes on the same chromosome, they show the linkage. And also crossing over produces recombination. Yes, of course. Here also some recombination is yeah, here also some recombination is occurred. And then recombination frequency can be used to determine the distance between the gene. If the recombination is more, the genes are far apart. If the recombination is very less, genes are close to each other. Yes. So what you should learn from this for the exam point of view is, that is a yellow and white. The recombination, yeah, it was 1.3 percentage, and it is tightly linked gene. And a white and a miniature, it is 37.2 percentage recombination, and it is loosely linked gene. Yes, children. Let's move on to the next one. So, a recombination and gene mapping. Recombination and gene mapping. So, what you can see here, say recombination. When we talk about this recombination, the formation of new non-parental gene combination. Non-parental gene combination. So, sometimes what happens, this linked genes, no, it becomes separated due to crossing over. Yes, sometimes what happens, the linked genes, it becomes separated due to crossing over. When it happens during the meiosis process, and because of this only new combination of genes called recombinants are produced. new combination of genes called a recombination are produced. And then what Morgan observed, see Morgan, he found that white yellow, yes, white yellow genes, it produced 1.3% of recombination, and white miniature, it produced 37.2% of recombination. So small recombination frequency means genes are very close, and large recombination frequency means genes are farther apart. Yes. So next we are going to see the gene mapping. So Alfred Stutevant. So what he said, see, he used recombination frequency in order to calculate the distance between the genes. So what he did, he used recombination frequency in order to calculate. See, there is a distance between these two things, and here also the distance is less, but still distance is there. Here also distance, he calculated the distance between two genes. So this only led to the development of the genetic maps. So because of this only, yes, calculating the distance led to the development of the genetic maps. So today, these maps are very important in genome sequencing, including our human genome project also. So what you should remember for the exam point of view is a high recombination frequency, greater distance between the gene. Let's move on to the next one. So let's have a quick revision. So dihybrid cross. So dihybrid cross studies the inheritance of two traits. And the parents, he can able to see the parents. You know the parents now R capital RR capital YY when it crossed with small RR small YY. So in the F1, so these are round and yellow, right? And what are the four gametes? These are the four gametes. You can able to see here. And what is the phenotypic ratio? It is a 9 is to 3 is to 3 is to 1 is the phenotypic ratio. And the law of independent assortment, each gene pair segregates independently. And the chromosomal theory, yes, the genes are present on chromosome. And Morgan, he experimentally proved that theory using Drosophila melanogaster. We saw that, right? And then linkage. What is linkage? Linkage is the inheritance of genes located on the same chromosomes, And recombination, it forms new gene combination through the crossing over. Yes. And recombination frequency is used for genetic mapping or gene mapping. Yes, we have come to the conclusion part. Watch the full video. Don't skip it, children. We'll see you in the next. Thank you.